Okay, we start? This is a really awkward setup. I have a computer to record the Zoom, a computer with my Linux stuff, and then to project it, I had to use this computer. So there's like 3 computers. So bear with me as I do. Like really bad, or maybe it's just because I need to be like moving the mouse. Let's see. It shouldn't go black as long as I'm doing stuff, I think, is my hope. Okay. So I'm going to do my best here. I'm definitely, will you close the door, Sandwer? I'm definitely, in terms of like coding and scripting, I'm far more comfortable with Python than I am with Linux. So I'm going to do my best here to just show you what I know from the perspective of a beginner. Hopefully that will be helpful because most of you will probably be beginners if you're first using this. So Linux is an operating system. It's come a long way. This is now what Linux looks like. This is like Ubuntu. So I have a computer that's dedicated to this. So you can just take like an old computer and you can delete your Windows operating system and you can install Linux on it for free. How to do that? It takes a little bit of time, but it's pretty easy. You can just Google it. You find instructions. You can just install it. The other alternative is, so one alternative is take an old computer. If you want to run a Linux system, you can take an old computer, put Ubuntu on it. The alternative is you can also download what's called a shell or like a virtual machine. So if you have Windows, you can download essentially like the operating system and you can run it as like a sub program on your Windows machine now. So it's a lot more user friendly. They've done a lot in the last, I guess, decade to make it more user-friendly to other people who are more comfortable with graphical user interfaces. So this is Linux. So I will open up what's called terminal. So sometimes you hear this referred to as terminal. Sometimes you hear this referred to as command line. Sometimes you hear this referred to as bash. Sometimes you hear this referred to as scripting. Scripting is essentially it's like telling the computer a series of orders of what to do. It's its own language in itself. So if you know Python, you might not know scripting or bash. Bash is actually the language that's used in the Linux system. So you might know Python, but you might not know bash, which is kind of like my case. Or hopefully you get to the point where you can integrate both because a lot of them are integrative. And oftentimes people will write bioinformatic programs in Python, but then to sort of like call them or to run them, you need to run them on a Linux system. So you kind of need to learn a little bit of both. So this is the terminal. I'll just kind of show you how it works. Again, this is a way of interacting with the computer that is not based on using the mouse, not based on using the graphical user interface. So the first key, like basic, super simple commands you need to learn are pwd. That's print working directory. That just means like if you're asking the computer like, where am I in the files? And tell me where I am in the files. So when I hit pwd and hit enter, it's telling me I'm in the folder of home. I'm recording on my iPhone, actually. So there's actually a fourth machine. It's ridiculous. It's complicated because my Ubuntu system can't connect to the internet. So blah, blah, blah. No, it's okay. Don't distract me. No, I'm kidding. No, that's fine. So like home/home, that's just a folder, and then /John, that's another subfolder within there. So you have to, that's the first thing, is like, if you just learn how to ask the computer like where you're at, that's pwd. And then ls is list. So it's essentially like in this directory of the folder of home, subfolder John, what are the files or folders that are in this, okay? So if you hit ls, which stands for list, it's just listing all the subfolders now within John, okay? So these are all subfolders. So most of my Linux stuff is organized on my desktop. So if you're gonna run stuff, you often have to navigate to the folder where it is, just like we kind of previously described. So the other command you need to learn is cd, change directory. If you hit change cd, it'll go, and you just hit enter, it'll go up a folder. If you want to go down a folder, you got to tell it where you want to go. So usually like the commands you're going to be doing is you do ls, and then you're like, of these, where do I want to go? I want to go to my desktop. So I'll hit cd, and then it's case sensitive. So you have to be very careful. That's a common beginner thing when you're starting is you try to do something and you don't realize it's case sensitive and you can't get it to work. It's because the capitals matter. Okay, so now I'm in my desktop. Let's do list. So now here's all the files, okay? So I see Python files, I see folders, I see files. Essentially, like, again, like you just want to get comfortable with navigating to stuff. There's more complicated commands, but I honestly don't think it's very helpful for me to teach those right off the bat. Like these are kind of like the three things you need to learn right away. Just remember pwd, ls, and cd. Okay. So this is how you would like run a file. Okay. Now let me talk about sort of like what's going to happen when you first open Linux and you try to do stuff. What's going to happen is you're going to have some plan. You're going to be like, oh, I learned about this like block of code that I could download and I could run. Okay. And all I need to do is just download it and run it, right? That's what everybody thinks. Typically what's going to happen every single time you try to do something in Linux is you're going to go to like the repository of those files. You're going to click on it. You're going to download them, okay? And you're going to get some folder. And then you're going to spend like a whole day trying to figure out how to like install it or get it running. It's always a huge pain if you're a beginner. So essentially like what's going to happen is you're probably going to go through a series of sort of like install commands where you need some certain packages. So you're going to see commands like pip, install, etc. So pip is Python install package. So you might, again, when people make code, sometimes they have what's called dependencies, where if your code is using stuff that other people made, but you don't have the module, you won't be able to run that code. Does that make sense? So as soon as you download something and you try to run it, there's going to be like 5 to 6 to 7 to 10 probably dependencies you're going to have to install. And typically what happens is you try to run something, and typically the error notations are pretty good. Typically they'll say like, I can't run this without this package. So then you just go through a process of typing like pip install whatever X package, and then you would hit like enter, and then it would install it. And if you ever have a question about it, again, like the best resource is just Google. Just Google like, oh, I need to have this dependency. How do I install it? It'll literally just give you a line of code that you can type into the command, and then you hit enter, and then typically it'll install it. So just know that like that's going to happen. Don't get frustrated. The really kind of one of the hardest parts as a beginner is literally just the first day of trying to get whatever you want running. That's the hardest part. So I have something that I want to show because it's a super cool program, and it's super useful, and I don't think it's something you can actually get on the internet. Like I think it's something you need Linux for. So if you have, say, a scenario where, I should be writing on the whiteboard. If you have like a scenario where everybody knows about BLAST, like you take one protein and you can blast it against like a genome. But what if you wanted to essentially take a whole genome of proteins called the proteome? So the proteome is all proteins produced by a particular organism. What if you wanted to compare like all the proteins of one organism to all the proteins of another organism to see if there were orthologs? So this is called like ortholog analysis. Find all the separate orthologs, find out the uniques. Like this maybe you're looking at microbes and you say you want to know this microbe has this set of genes, but this microbe does not have this set of genes. So you want to know like how they're different. That's a super common bioinformatics thing, and it's at a level of, I guess, bioinformatic knowledge that's above sort of all the basic stuff that I've taught. So I'm going to try to show you how to do that. So the program we're going to be working with is called FASTA36. Okay, so I have previously, oh gosh, it's going to update. I don't want to update. Do I hit cancel? Very good. I just want to open FASTA36 settings. So as you like go through here's one of my best advice. Again, I'm not the expert at Linux, but I know the frustrations you're going to run into because I've run into them. So what you want to do is whenever you have like a program that you want to run, you should take very detailed notes about, as you start the process, like what you're doing. Okay. So for example, when I got this program running, like I wrote down essentially like what the program was, where I downloaded it from, the website, and then recall back to the computer science lecture where we were talking about methods, where methods are going to have parameters, and you can't run a method unless you know, the parameters in that method. Bash and calling like programs like that is very similar in a sense that if you look at this command, this is the actual command that when I finally figured out how to run everything I wanted, how I wanted to do it, this is what the command looked like. Okay, so it's a pretty complicated command. And we can just sort of dissect the parts of this as an example of what you're going to encounter. So FASTA36, that's essentially like telling Bash to run this program. Okay, it's the same thing as if you were to see when I ran the bot, I typed Python something like main.py. It's similar to that in a sense of you're telling the command line, run a Python file, this is the file. This is kind of a similar thing where you're telling it you're going to be running a FASTA36 program, and then you start to get into the parameters. Okay, so here's one thing where people get too intimidated and they kind of give up, which is if you're looking at this code and you're like -M8, -B1, and then a bunch of random stuff, it's kind of like really confusing and really intimidating in a sense of, I have no idea what those mean, nobody's going to understand what these mean, this doesn't make any sense, and you're going to feel that way. But you need to just understand that these are parameters, okay? And probably what they mean is probably in the documentation of the program you just downloaded, okay? So here I wrote down, as I was figuring out these parameters, I kind of like, again, you would want to write down and take notes of what the parameters mean and what your options are and how they change it. So if you type within FASTA36, again, you don't need to memorize this, it's just an example. Within FASTA36, if you type this -M, it's saying that it's going to do like a mimic of a blast. And if you type -M8, it's going to give me this special tabular output format, which is how I wanted. So it's essentially like when you call the method, oftentimes you need to tell it what to do. You need to tell it how to specify the output. And you figure that out through a process of just like reading the stuff, trying things out slowly, trying to run stuff, seeing what happens. Okay, the other parts of this input are, which is very common, is you're often trying to locate like source files. So remember how methods need inputs and then they generate an output. Essentially, these three parameters here are two pieces of input. So here's a file, a FASTA file. Here's the location of it. That's one input. So remember, if we're going to blast two genomes against each other, you're going to have one genome, like literally a file with one genome or a proteome. And then you're going to have a second file with a second proteome, okay? So you need to tell, essentially, like the system, here's where this file is, here's where this file is. And then it essentially runs the program with this, it runs the program with this, and then it spits out an output file where you tell it where you want it to go, and then you can name it. So I could actually like type whatever I want. Like I could type like, this is an example output. And if I run it, like hopefully this file should appear on my desktop. Oh, wait. Oh, I'm not in the terminal. So if I run this. Also, copy paste is different in the terminal, which is super annoying. So it already ran, and let's see if we can find the file on my desktop. Here's the file it generated. So again, some of these things are weird in the sense that sometimes when you run it, you'll get like, oh, it's running, and you'll get like, no, it's like, and you know it's running. Other things you'll like run it, and it will just be like, burp, and you are kind of like, what, did it work or not? But here's the output file. But before we look at that, I want to show you how you would do this, because this is a super useful tool. Okay. So the first step would be you need to get the input files. Okay, so here's what they look like. Let me just show you what this is the output. So this is, what do you think this is, just by looking at it? What do these look like? Yeah, so they're proteins, right? Mm-hmm. So this is literally a proteome from, here's Wolbachia WML. So proteome from or it might be WPIP. And it's in FASTA format. So remember, FASTA format is a caret. You'll see some names, some stuff, and then a carriage return, and then the sequence, the amino acid sequence. Okay, and it's literally just a list of a bunch of FASTA format proteins from, it's essentially every single protein that's encoded in the genome of WPIP. So there's 8,000 lines of essentially like coded content. Okay, so again, like you could never like look at this and blast these individual genes individually through the blast thing. That would take forever. It'd be impossible. So let me show you first where you get this from. So you're gonna go to, guess where you're gonna go? Hopefully my internet works. Guess where you're gonna go? Really? That lost? To get the FASTA files. There you go. So if you go to NCBI, so you can search genome. Let's say we wanted to find a Wolbachia, which is a bacteria. Or what bacteria do you guys like? What's your favorite bacteria? E. coli? Yeah. Okay, E. coli. Let's do E. coli K12. That's typically like a lab strain. So we should be able to find the genome. So here's the genome, like the accession. Typically, there's a button to click that will allow you to just literally just download the file of all the translated proteins from this organism. And I think it's this button, download sequences in FASTA format for genome or protein. So I'm gonna click this and see what happens. So I just downloaded something. And this is what it is. So it's literally like the same thing that I just showed you. Like we just downloaded one. That's how easy it is. That's how easy it is to download every single protein from a particular organism. Now you could imagine now how the little information I gave you about Python might be useful, right? Because if for some reason the format of this downloaded file was not in the right format for what you needed to run for your Linux thing, you would need to adjust the format, okay? And so one way you could do that is by manually typing, or if you were good, you could code a little Python thing to just quickly run through it and parse it properly. So you can understand why that would be important. Okay, so I showed you essentially like how to download the genomes or the proteomes. So now let me show you the two that I have, and let me kind of discuss like the experiment that I was running. And if anybody has interest in coding and you want to get a paper, we could easily get a paper with this. But I haven't found a student who's been willing to do it yet. But, okay, so for example, let me give you the thing I would like to study. So Wolbachia is very difficult to culture. Okay, it's an obligate intracellular pathogen. It has a reduced genome. So that means it's living in a host cell, and it's often like stealing food from the host. So it's, in many circumstances, is lost parts of its genome that were essential for it living on its own. Okay, so if you ever wanted to culture Wolbachia like outside of cells, like in like a media, like E. coli, you could imagine there might be a scenario under which you could derive a certain like recipe. And if you just concoct this magic recipe that's giving it all the nutrients it needs, you could then perhaps like culture Wolbachia in media, right? Does everybody kind of get that gist? What would be the first step to trying to figure out what Wolbachia needs in the media?
What cells it is what somatic cells or germ cells they're in the insect. You need to know like what host they're in.
Okay, well, we know that. We know it lives in the germlines of mosquitoes or insects.
You know like what nutrients are provided within the germ.
So one way to do it, you're saying, would be like to analyze all, essentially like make a list of all the components in like the cytoplasm of a cell. But does Wolbachia need all those things?
No.
So I guess my more question is,
Analyze its genome too for anything like that codes for metabolites for those things that are found in. So you'll need both.
yes. Okay, so I like the second answer better, which was if you just kind of like look at the Wolbachia genome, you're going to get a hint about what it eats, because you're going to find genes like, if, for instance, if you found a LAC-Z gene, you'd be like, oh, it likes lactose, or it can eat lactose. Same idea. Okay. So that would be like a first step. Okay. To sort of make it a better guided experiment, you would want kind of like a control, right? You want to compare it against a control. So what's a bacteria that you know that can easily be grown in media? E. coli. So what might you want to do? Yeah.
Compare E. coli and Wolbachia and see like what genes E. coli has that Wolbachia doesn't. Because
Yes.
those are probably like the things that like Wolbachia doesn't have them because it's not
Yes.
like extracellular.
Yes. So to reiterate, very good. You would want to take something that you know you can culture, compare it with what you can't, and see what's missing from the thing that you can't. Okay? And then either there'd be a few things you could conclude from that. Maybe you could change Wolbachia by adding in the things it's missing, or maybe it depends on how big that list is, right? If it's like 1,000 things, it's going to be really difficult. If it's like 1 or 2 things, you might be able to just add in the genes. Okay, that would be one approach. So that's essentially kind of like what I want to do is, okay, my idea was I actually don't think Wolbachia can be cultured. And the reason I don't think it can be cultured ever is because if you take the Mycoplasma minimal cell that was created by the Craig Venter Institute, that one has, I think it's a mouse moving thing. That one has about 300 genes, okay? So there's only about 300 things you need to be able to live autonomously in some kind of a media. So I kind of thought like the best way to do this would be to take the minimal mycoplasma, 300 genes, compare that against the Wolbachia genome and see like, okay, of the 300 core things that everything needs to survive, what is Wolbachia missing, okay? And when I did that, it was missing like half of the stuff, like 150 genes out of the 300, like it's missing. So essentially like my conclusion was you probably can't culture it because there's probably not a way to add all those things. Anyway, that was kind of like one idea. Like that's an actual like research example about why you might do this. And like I said, if anybody wants to get a paper out of this, You could do that. You could write that up. So let me show you then now, like once you understand that, like the setup of the premise, why you might do this. And it doesn't just have to be about Wolbachia. It could be like whatever. There are so many applications of this type of analysis for your own research system. All you need to do is download the 2 files. So in this case, I have a FASTA file from WML, which is a Wolbachia from Drosophila melanogaster, and I have a Wolbachia from WPIP, which is from Culex pipiens. So in this case, I'm comparing 2 different Wolbachias, but you could easily see how, depending on the experiment you want to do, you just download different genomes. Okay. And then you literally, like I did, I literally just typed in the command, I hit enter, and it ran the analysis, and this was what the output looked like. So essentially, what do you think these are? Yeah, these are the accession codes for the proteins that it's looking at. So if we copy this one, and we went to NCBI and we searched protein, this should give us a specific Wolbachia protein. So it says anchor and repeat domain from Wolbachia endosymbiont of Drosophila melanogaster. That was literally this first one. Now, if I search this one, this second accession, I'm guessing this is the ortholog it found in the other Wolbachia. So this next one should be a hit from WPIP. Yep. So now this is this exact same protein ortholog from Culex pipiens. So now you can kind of see now, like we understand how to interpret the data. It's saying this protein matches this protein. Somewhere you'd look and you'd find what these are. These are probably scores. This is probably the BLAST score, 1.7 e to the minus 26. That's a good score. I think the lower it is, the more likely the alignment is. And you can essentially, like, you can format these outputs with the parameters to give you what you want. So if, in this case, all I'm generating is orthologs, which is the match from here and the best match from here. Okay, but you can easily like reprogram the FASTA36 to give you unique genes. So in that case, it would do a blast of the entire genomes, but it would only give you, it'd spit out things that were totally unique to the one organism versus the other. And that's what you'd want if you were doing like the media question. That went pretty fast. That's kind of like it. I don't want to I can't teach you guys how to code, but my only hope is that this is sort of like reducing the fear of starting something in Linux or trying some coding. It's really actually pretty easy. The hardest part is just getting stuff installed. Are there any questions, or do you want me to like try anything or show you anything? Or is that's kind of it?
Can you download something and then show us what we can
That's
expect to see when we?
That's a good idea.
Dependencies?
Yes. Let's try to find something like to download. What would be? Do you have any tools, Cassidy, that you use for like Bash or Linux?
To do what?
He wants me to just like download something to demonstrate.
I mean, like, it depending on if you know what you're trying to download, like, we use like SRE toolkit, which is like
SRE toolkit?
SRA toolkit.
SRA toolkit.
And then like you can access and get the FASTA files for like SRA, like sequence root archive.
So okay, so this is like a Linux thing?
Well, it's NCBI, but then it's like a command line tool.
Okay.
For SRE toolkit.
Oh yeah. Let's see. Okay, let's see. Now this is where the dumpster fire will really start.
I'm like.
So, but it is good in a sense of like, so here what you're seeing is, so just when I Googled that, like you're seeing a guide, like that's always where you start, and there's always going to be like install instructions, and you have to kind of like look at those. Let's see if I can find down.
I was lucky in using this because we're running it all on the Algebra supercomputer. I
Yeah.
already have it, so you just have to load the modules and stuff.
Yeah.
So like the setup time for that is really different than like downloading it and setting it up yourself.
Let's just try to
Yeah.
hang on. Actually, I I was downloading like transposon ideas, like when you were, that was a while back.
That was a while back.
What's something, actually, like, let's just think of like, what's something we might want to do that we could like try it out? Like, we could try the transposon thing. So one thing Kyle and I were interested was like, is there any pre-made software that can just quickly like identify inverted repeats? So it'd be something like transposon inverted repeat finder, bioinformatic, Linux. Let's see what comes up. Mite tracker, an accurate approach to identify miniature inverted repeat transposable elements in large genomes. So let's see if we can like download this. Mite tracker. Download Mite tracker.
Remember that one that we tried like a long time ago? It was like 4 hours. And we
Probably.
There's also a web-based like toolkit. It's called Galaxy.
Galaxy?
Use Galaxy, I think .org, and they have like SRE tools. They have a ton of stuff that's like on there. You can use it on your stuff. You just upload your data, and I can use all these tools.
Or you can like do it on the internet.
If
Yeah.
you don't have Linux.
Yeah, so that's often like the easiest way if they have like a web server that can do it, but oftentimes you can't do that. So typically what's going to happen is you're going to end up at this website called GitHub. GitHub is like where people who code, they will like put their code so you can download it. So I'm assuming this is the GitHub site. This looks funny. This is the GitHub site for these people who are making stuff with transposons. This is a strange user interface.
It's because it didn't load correctly.
It didn't load correctly?
You have to reload it. Yeah, it just didn't load correctly.
There we go. Okay, so for example, you're going to see something like this. So anytime you see a .py file, what do you think that means? Yeah, it's a Python file. So like that already, you kind of like know a lot more than other people who would like literally know nothing. The other thing is, so when people are assembling pieces of code, when you're building like things that are integrated, there's typically like a main file, which is called the driver, which is, that's the thing like that runs the program. So in this case, it looks like they've built a subfolder for results. It probably spits results in there. It looks like there's a subfolder for TMP, these other things. But one of these is probably the thing you got to run the program. So it's definitely not this. This is the README, that sort of like where you'd go for the instructions. It's probably one of these PI files, and it's probably this thing. Okay.
I'm going to find IR that's finding inverted repeats, so it's probably that one.
You think it's this one? Okay. So what we can do is, the first thing you would do is you would figure out how to download this. So you just download the zip. It's going to go to my downloads. I typically on Linux, I would put it on my desktop. Like it's just easier for me to know the things I'm working with are on my, maybe I need to unzip it. Extract here. Why is it not letting me do that?
You should have extracted it to your desktop.
What? Let's see if I can just paste it. Is it literally like not working? Paste to copy. Would you say extract to desktop?
Yeah, you can extract too.
Extract to desktop. There it is.
Oh, you had it.
Good.
It worked the first time.
Oh, did it?
Yeah.
It did it twice. Okay. So then you'd have it, right? So now, like, okay, so here's exactly what I was talking about. We're in the situation where we've got this tool. We envision using this tool. We've downloaded the folder that has all the files. It's probably got some of the dependencies. Now the goal is to figure out like how to run it. So the next thing you'd probably do is you'd probably look at the README. There we go. So you'd look at this README. It's saying like, it's talking like about it, requirements, installation. So it looks like these are some commands. It's saying like, so for example, in order for me to run this, like a prerequisite is I'm probably going to have to download these sort of dependencies. So let's try to do this. Wait. This I think we already got. This was literally what I just did. So here's the other thing with when you're trying to install this stuff is sometimes if you're just not familiar with computer science, like sometimes you'll end up doing things like multiple times because there's different ways to approach it. But okay, we already did this because we cloned the repository. That was when we downloaded the file. So the next thing would be essentially it's saying blast, this program is a requirement. So we got to install this NCBI blast virtual environment. So this is good. And this is, you can tell that this is a bash script argument. So then what I would do is I would go to my bash and I can type that in and let's see what happens. Oh, it's gonna ask for my password. That's it, so it's working. So do you want to continue? Yes. So now it's downloading. So you're gonna have to go through like a series of these sort of like installations to install the dependencies. I don't know how long this would take. I won't make you guys sit here forever, but like you get the point, right? Like this is how you would approach it. And then once you start trying to run it, like even I guarantee you, even once you get through the README and you've done all this stuff, you'll try to run it and it'll say it won't work because you don't have this module. And then you have to search how to install that module. Won't work because you have the next module. And you just progressively go through until you get closer and closer and closer to actually running the program. And then you can actually run the program at the very end. Does that make sense? Okay. That's kind of it. I wish I was like a bash expert, but I'm not. But I do think it's important to expose you guys to this. Okay, so I'll end it.